Well, hello, everybody. Thank you for coming. Welcome. We are going to be having a kind of a cool webinar today. I like it because I'm not presenting it. So we're going to hear from three amazing biotech teachers. They are going to be sharing their experiences as they built their biotech program, what they're doing now, some struggles and challenges they've had, some successes. So all that cool stuff. So this is going to be a really nice opportunity for those of you who are thinking of starting a biotech program, or perhaps you already have a biotech program and you're just looking for more ideas and information. So let me go ahead and get y'all started. So for go over the agenda real quickly. The first thing that we're going to do is give you an introduction of everyone. We'll have a panel discussion where I kind of go through and we have some questions that we've already put out there for our teachers to answer, and they've put their thoughts down. So we'll let them talk. As they're talking, I would love for you to get in the chat. We have Yolanda here, who's part of our marketing team. She's awesome, our website team and all that good stuff. So she's going to be monitoring the chat. She can pause and let us know that there's burning questions that you want our teachers here to answer. So please feel free to put that stuff in the chat. We can always pause the discussion if it's something that's relevant to that particular question that we're answering. At the end, we'll also have time for more Q&As. So if you have some questions that we're not really hitting, but you're just dying to know what they think, please save that for the end and we'll answer them there. And Yolanda, I guess if you have some that you think would be better at the end of the session, if you can kind of put those off or just let them know that we'll add that to the Q&A at the end. We'll leave about 15 minutes at the end for Q&As. And finally, at the end, I'll show you some resources and things like that. Okay, so here we go. Just to start off, my name is Lee Brown. I'm a curriculum and training specialist at Bio-Rad. Although Bio-Rad is based in the Bay Area of California, I live in the middle of Texas in New Braunfels, so just between Austin and San Antonio. I'm a former teacher, but I've been working at Bio-Rad now for 13 years. And my role is mostly to train and work with teachers in biotech, biology, all kinds of life sciences, really, from typically high school to university level, and help them bring these biotech labs into their classroom. So I've worked with all of these teachers that we're talking to today. And if I can help you with anything, we'll have a survey at the end, and I can you can reach me that way, or I can reach you that way. We have with us Sherry, Jamie, and Michelle, and I'm going to let them introduce themselves. So Sherry, take it away.
Hello, thank you for being here. My name is Sherry Anne, and I teach at Brebeuf Jesuit. I've been teaching 32 years, with the most recent 27 years at Brebeuf. I created a biotech course in 2003. The course is a science elective. My school has 800 students, 9 through 12. About half the senior class takes my class, and several double up in other courses as well. My course is a full year course, and I'm past president of NABT.
I'm Jamie Allison, and I teach at Loveland High School in Loveland, Ohio. That's just outside of Cincinnati. I've been a teacher for 36 years, and I've been doing the biotech thing for give or take 18. The program is honors. We have 4 sequential classes, 22 students max in each class. Michelle?
Michelle, you there?
She muted.
I am. I was muted.
There you go.
Back to online teaching days.
Right.
Hi, everybody. I'm Michelle, coming to you from not so sunny Phoenix today, but it is 77. I have been teaching. This is my 27th year in the classroom, but for 17 of those years, I was a general biology teacher. I didn't even teach any honors level things. And then for 14 years now, I've been teaching biotechnology. In the state of Arizona, we call it bioscience, and our standards fall under bioscience. So I am a career and technical education teacher, which means that I have the certification and some industry experience, not really industry experience, but I've had internships and had relevant experience to get that certification. So I'm also a HOSA advisor, because in CTE programs, we also have our career and technical student organizations. And this is my 14th year as a HOSA advisor. So students in our program, it's a very unique program. We've taken 2 years of standards state standards and turned it into 6 different courses. Students actually have to apply to our program as 8th graders, and then we have them for 4 years through that series of 6 classes. So our high school is on a block schedule that really is to accommodate it. We have computer science and engineering pathways also for students in our program. So I teach in a laboratory. That means that I have a maximum of ideally 24 students, but I do have 26 in 2 of my sections right now. And so I teach 4 different preps every year, but it's 2 preps a semester. So it's really nice. Our classes switch over at the semester time. I think we have a little bit of everything here tonight for you, different flavors. And so hopefully that will resonate with all of you.
Yeah. So it's a wide experience here. So first question is, how did you in teaching biotechnology? What kind of sparked that interest, and what motivated you to pursue a career in biotech education?
So I will go first. I actually interned at our Marion County Crime Lab, that's in Indianapolis, in 2002. So if you think about 2002, CSI was really hot. And so I was in the Marion County Crime Lab, and I was working for Dr. Mohammed Tahir. This is the very first day I walked into his lab. And if you look carefully, I've got a zoomed picture, he's filing a bone because he's trying to determine, to extract the DNA from that. Now, what's interesting is Dr. Tahir was the very first person to extract DNA from cells on the back of a wristwatch, chewing gum, and cells from a toothbrush. Now, keep in mind, this was the summer of 2002, and we know what happened the previous fall, right? September 11th. Well, because of Dr. Tahir's work with toothbrushes, he was flown out to New York City, and when the towers fell, unfortunately, in many cases, the remains were so small they couldn't identify people. So then people would bring their toothbrush, a known sample of their loved one, and then Dr. Tahir was instrumental in bringing closure to some of those families. He also worked on the Sam Shepard case. I'm 54. If you're my age, you know Harrison Ford, The Fugitive. And so Sam Shepard was the fugitive. So he helped bring closure to that as well, exonerate Sam Shepard. And then this was the first time I show my students these pictures. This is the first time I saw a DNA sequencer. I don't know what's happening. It seems to be. Oh, there we go. DNA sequencer. So I took a picture with my big camera back then. I did not have a cell phone. And then the first time I saw a thermocycler. So again, this is 2002. This is really exciting to see a thermocycler. So then I'll tell you a little bit more about starting that class coming up. I'll turn it over to Jamie.
How did you get started in teaching biotech? So
I was brand new at the school I'm currently at now, and I did not have a hook. And the principal said, hey, your certification matches this program we're thinking of doing. You want to get in on it, go to a couple meetings, see what you think. And if you think it's a good fit, went to the first meeting, didn't wait for the meeting to end, sent the principal an email, said I want in, and the rest is history. We ended up writing the curriculum for the whole state, and I haven't looked back since.
That's amazing. Uh-oh, hold on. I went the wrong way. Michelle, how did you get started?
I never intended to be a biotech teacher. As a matter of fact, I really didn't know what one was. I was teaching general biology. I had a department chair that said, I want you biology teachers to all start incorporating some biotech into your classes. And here's a workshop that you can go to. So I actually went to three different PGLO workshops before I had the nerve to actually do PGLO with my students. And then she shoved an application at me and said, the state of Arizona wanted to train biology teachers to be biotech teachers. And she said, fill this out and you need to do this. And I was like, okay. And I'd never even used a pipetter or gel electrophoresis. I couldn't put a chamber together. And that was then. I started taking classes, loved it, got my certification. I will never go back to teaching general biology.
That's awesome. And here you are now. Yeah, we, for those of you who don't know Bio-Rad, we do workshops all over the country, typically at science shows and also local state shows. And so a lot of groupies that come and do the lab, over and over with this, because it's hard to do it after just seeing it once. So We love that. Okay. Next up, how do you structure your biotech course?
So
So,
in my
Sherry?
case, yes, in my case, I say I do labs, labs, and more labs. I've gotten to the point, and you're going to see some of our history here as we go through this in starting our biotech courses and so on. Anymore at this point, if the students can't have an actual hands-on experience, a wet lab, then it doesn't make the curriculum. There's just so many things we can do that, again, if we don't have a wet lab experience, it usually it just doesn't make the curriculum. I do a lot of use of models and manipulative, so students have a model, a mental model of what's happening at the molecular level. Everything is done within the context of a real world scenario. Now, if you order from Bio-Rad, most of their labs, if not all of them, are written in a real world scenario, but I use a lot of case studies as well.
Nice.
And I'm going to go through some of the labs that I do in a little bit here.
Cool.
So for me, the structure is really simple. We're on a 7-period day. I have first and second period blocked together for my juniors. I have 6 and 7 blocked together for my seniors. And we switch actual numerical classes at the semester. So we have the 4 classes, bang, 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 and they just matriculate through. Once you sign up for the program, you've made a 2-year commitment, 4 classes.
Nice. Michelle?
So we have a lot of students who come into our program and want to go into primary healthcare or to go into research. And so we actually, as I said, we're on the block schedule. So we have 82-minute classes every day and then do a complete class in a semester. We also keep lab books with the students. We make them document everything in a professional lab book. The first year they format them, then we actually buy them hardbound lab books that they keep. So we are really kind of into the documentation, but like what Sherry said, labs, labs, labs. We do a lot of labs. I could count on one hand the number of times I actually do anything that even closely resembles a lecture during an entire course, because we just try to give the students modeling problems, activities where they're thinking, they're collaborating, they're problem solving. And when the labs fail, awesome, that's real science. And we ask them to
Indeed.
figure out what happened.
Yeah, that's a really good point. One of the things I didn't think about in here, but I know is probably a question that you've probably addressed, and I know other teachers out there are probably curious about. For short classes, I don't know if I've ever done the 50-minute, 45-minute classes. How do you make that kind of thing work in with the labs? Have you had, or have any of y'all done that with the shorter class periods?
I used
That's
to
a
teach
good
in a school where it was 50 minutes.
Yep. And so how was that? How did you make the labs work in that amount of time?
Really prepping them ahead of time the day before so that they know exactly what they need to do when they get in for a lab. And then for some of the longer labs, and Bio-Rad and other companies will often build in breaks and say, if you need a stopping point, stop here. So if you know ahead of time that you're not going to get through something, know where your stopping point is. And that's really helpful to have that kind of planned out ahead of time.
Yep. Yep. Awesome.
I found
Thank
setting
you.
up PCR reactions
Yeah.
on a 40-minute period works. DNA
Yeah, setting up.
extraction, you're probably going to need 50 to 60 minutes, depending.
Yeah, it's kind of a trial and error thing, right? Because some things you can do in just a few minutes and get them started in the thermocycler, and then others are going to be more than 50, so that gets tricky. Okay, how did you start your program, and how has it grown or changed over the years?
In 2003 was a very special year for me. One, I gave birth to my only child. His name is Anthony, and you can see right from the beginning, he loves science just like his mother. So this was June of 2003, and I mentioned that I interned at the Marion County Crime Lab in the summer of 2002, and I was so excited about it that fall. My principal said, "Why don't you start a class next year?" And I said, "Okay." And I was not pregnant at the time. I'm like, "Sure, this is great." So then, I was pregnant, and now it's March, and she's like, "Sherry, you sure you still want to start a new program, a new class in the fall, a month after, your child is born?" I'm like, "Of course I do." And it was 4 sections. that was a full teaching load for science teachers at the time. And there I started a biotech program with just a little bit of equipment. So she said, what's the base? What do you need so we can see if we can do this? I said, I would love a thermal cycler, which that seemed pretty extravagant at the time in 2003. And it was, but I was able to get a thermal cycler. This is not one that could refrigerate your samples. So I had to be there until 7:30 at night, and did that for many, many years. I asked for some gel chambers. I was able to get a set of 6 of those. For some reason, this is stalling. And then I was able to get 6 micropipetters, and I chose the 0.5 to 10 because then I knew if we had to measure 50 microliters, we could do it 5 times, and indeed we did. But I got a set of then 6 of the smallest volume. I had a microcentrifuge, I had a water bath, and a vortex. And now through the years, I've been teaching, this is what, my 21st year, is that right? So I've accumulated equipment as I've gone. Sometimes when, well, next year could I get a another centrifuge? Next year could I get another set? Can I get a different volume of micropipetters, etc. And I tell my students, I say, you are the beneficiaries of 20 years worth of students taking such good care of this equipment that you're lucky to have such I think it's pretty robust now what we have. But again, it was gradual. I'm still using the same that blue micropipetter. I took a picture of that yesterday. That's one of the originals we still use. Now, here's the thing. I mentioned that I interned at the Marion County Crime Lab in 2002. Well, our school newspaper, the spring where kids were signing up, so this would have been the spring of 2003, knew that I had done that, and they wrote an article saying that CSI is coming to Brebeuf, a new class. Whereas, I was calling it biotechnology in the newspaper, school newspaper, it said CSI. So again, I had all these kids sign up. I had a full section. And so that school year, they kept going, when are we doing CSI, Miss Honey? When are we doing CSI? I'm like, this is not a CSI class, it's a biotechnology class. And they just kept, I'm like, okay, I will do this at the very end of the year. These are seniors. I had a lot of them as freshmen. I said, we'll do this. I said, but I want a student committee. You will devise, volunteers, you'll devise the scenario. And it's always someone fictional who dies because I don't want kids walking down the hall going, oh my gosh, our principal died. And then, freshmen are hearing that. So it's always someone fictional. And so the faculty staff serve as the suspects. The kids go out and interview them, get DNA samples, fingerprints, and so on. And I'm telling you about this now because I'm still doing it. It's a project now that has lasted every single year. There's no way I could ever end it. Sometimes the principal will bring people around to my class and say, oh, this is the class that does the CSI project. And it's like 2 weeks at the end of the year. Anyway, so it's a lot of fun. It's a way to bring the community together at the very end of the year, and seniors just have a blast. AP exams are going on and they're solving this mystery. Here's examples, and then they end up arresting somebody. So they have a lot of fun doing that.
Oh, that's
and
awesome.
It's always in a public place in the building. So, okay, that's it for me for now.
Jamie.
So my program was not the first in the state of Ohio. There were a small consortium of schools just north of us in Dayton that were kind of doing things, but they really had kept to themselves at that time. So the Cincinnati Consortium, we had 4 or 5 high schools, and we had 3 college partners that were involved. And we put this whole curriculum together. And essentially, we're teaching that same curriculum today. What I have done is incorporated new technologies to adjust with the times. So as CRISPR has gotten more and more popular, CRISPR had to be part of the curriculum. As RNAi kind of was a flash in the pan in the midst of all this, and now you can't find anything about it. So we've kind of let the RNAi go. But essentially, we just started this little group of us, and most of us are still kind of doing it somewhere around the state. And we just changed with the technology.
Nice.
I've had 2 different experiences. The program that I'm in right now, I came in as a second biotech teacher, and that original teacher has moved on. And a shout out to my teaching partner, Amanda Cherry. She's on this webinar tonight, and Amanda is also a Bio-Rad fellow. But before I came into this program, I started a program in another school district, and I had a meeting with the CTE people and my principal, and they're like, "Awesome, yeah, this is great, awesome. We'd love to have this. We don't really have any extra money for you." And I know we're going to talk about funding coming up, and I did write some grants. I'm like, well, what is my most important priority equipment that I need? And I was able to find some funding sources through grant writing. But the program that we have in now, like with Amanda and I will sit down, and we always at our PLC meetings, we talk about what are some recent things that we've seen. Would this particular lab or content be better in a one of our other courses? So we're constantly evaluating, looking for new things, seeing what other things we can retire. And that's just, I think, something that, because biotechnology is always changing, you just have to find out what in your area is a priority. Are your kids going to go directly into industry? Do they need to go to community college and get some type of certification? Or are they looking to go to a 4-year institution and beyond? And really tailoring your program to what your student population is really interested in and to what maybe might serve the greater community is really something to consider as you start a program.
Absolutely. Yeah, a good mix of you'll all have a good mix of changing things up, and then keeping those really popular labs. CSI, it's always a hit,
Hi, Lee.
Okay.
We do have a question in the
Yeah.
chat about funding. Are we covering that
We will
later,
cover
or should we
funding.
go with that now?
Yeah, we'll cover funding in a little bit. Yep.
Okay.
Great question. So keep that thought. Okay. Are there any specific topics or labs that are particularly engaging for your students?
So I'm going to go through several here, and all I want to really do is mention it, give you a brief synopsis of it, show you pictures of students engaged with the lab, but I will not go into any depth about these. We don't have time, but in the Q&A at the end, if you want to ask me about a particular lab, or Michelle or Jamie, I'm sure if they've done these, they'll jump in as well. Okay, so restriction digestion analysis of lambda DNA. I know some people don't do restriction enzymes anymore. I still do, and I'll explain why, but you can see here students working with the samples, and then here's a sample gel. Now this is a sample where I've stained it with the blue dye, so the staining overnight and the destaining, and you can see fairly nice results there. Then PGLO, and the reason I do restriction enzymes is because PGLO, then I want them to understand how that recombinant plasmid was made. And so we have really nice results every year with PGLO transformation, as you can see. And then I do the follow-up with the GF purification kit. A student actually took this video. It's fast time lapse. Yeah, so you can see the GFP under UV light once they add the buffer to release it coming through. And so that's a really nice lab for column chromatography.
Oh, that's a cool one. I love that. There it is.
So there you see, yep.
Nice.
And you can follow up the SDS page. You can run a vertical gel as well and look at the bacterial genome. I am trying to fast forward. Okay, the GMO investigator kit, my students were just working on this last week. So this is where they extract DNA from different types of foods. I say we're going to do the O's food group: Doritos, Fritos, Cheetos, and Sun Chips. And then we do some things that claim to have not been genetically modified, like Skinny Pop. We do a meatball because there's soy filler. Has the soy been genetically modified or not? But everything we do, we're testing for plants in this case. I buy like Panda Puffs. Oh, and there's a fun, if you've seen blue tortilla chips, there's a brand called Garden of Eden, which of course is spin off of Garden of Eden. And they've not been genetically modified, they claim. And so we test for that. And so you see students here in the lab running their gels. And then I want to show you, these are pictures of gel results that I've gotten in class. Okay, so then the PV92 PCR informatics kit. My kids are doing this week. These pictures were actually taken yesterday. Working on the lab, loading the gel. And then in this case, like the last gel, used the U-View. So you can view it under the transluminator. You can run a 10-minute, 15-minute gel. So my students loaded the gel, and then we went back into the room to talk about what we might see on the gel, and then come back, and with the U view, you can see your results within the end of class. A 60-minute class, you can run a gel and view the results. Okay, fish DNA barcoding kit. The way that I use this kit, and I started this in 2014 using this way, I tell my kids I'm going to take one for the science team. I go to local sushi restaurants and I order a nigiri, and I eat one piece and then bring one back to the lab. And so then we extract the DNA, and they then amplify a certain region of that. And then we run a gel to see if indeed they were successful with that PCR. And if so, we send it off to get sequenced. And then we get the sequence, we put that into NCBI or GenBank, and we can see if the fish is mislabeled that I've eaten. Every single year I've eaten mislabeled fish. You can imagine students love this. At this point, I've accumulated quite a record of restaurants in the metropolitan area of Indianapolis. So I try to always go to different restaurants of what they're serving and what the results were. And that
Sherry,
really
what's the most commonly substituted fish, or the one that pops up the most often?
Yeah, if you see a restaurant serving red snapper, it's likely tilapia.
Tilapia. Yep, that's what
I went to a fancy restaurant in Indianapolis last year because they were serving red snapper, but it was like a fillet, and it was $42. So I'm like, I bet this is red snapper. It was. But then I went to a fancy place in Detroit last year when I went up there for spring break to see Bruce Springsteen. But anyway, and it's like a fancy place, but they were selling their nigiri of red snapper for like $4. I'm like, I'm not sure. And indeed, it turned out to be tilapia. And I called them, and very quickly, our communications person came to me a few years ago and said, Sherry, if you're willing, there's a local news station who wants to run the story, but only if you share the names of the restaurants. And so I said no. I said, but let me talk to my students about this. And they said, my students are like, yeah, go ahead. And of course, they've lived, right? They grew up in Twitter and all that. Like, you tweet things out and you try to get likes and so on. I said, no, let's reflect about this for 24 hours and come back and talk about it. And we had a great conversation, and they decided we ought not do that for a lot of reasons, but I don't want to take up too much more time. So anyway, here's the virus detection transmission kit, new last fall. It's the Science of Opioid kit, which still exists, but repackaged in this way. Phenomenal, because students can look at the chain of infection. They love this, and you're going to see me talk more about this in a few minutes. And then we run an ELISA. I do it with the giant panda problem. You're working with real antibodies and real antigens, but in this case, you're trying to see if a female panda, there are only 1,800 in the world, females only ovulate once a year. Is it ovulating or not? And then it's really nice because this is Bio-Rad. They've got some manipulatives, some paper where they can show their understanding of an ELISA. And then because the kids loved it so much, Chief, I'm the caretaker of our school dog. He's a French bulldog. We dressed up as pandas for Halloween, and you can imagine the kids love that as well. Okay, that's
awesome.
Oh, okay. All right, we do a Western blot. Okay, I'm gonna stop. Yeah. So we do the Western blot.
That's awesome.
Backwards. There we go. This is Michelle.
Michelle.
You're muted, Michelle.
Yes, to all of the labs that Sherry has talked about, and I teach a microbiology class in the fall with the seniors. It's our capstone course. We just did that virus detection lab, which I know Sherry was being modest and helped create the curriculum for that. My students loved it. We did it as part of our epidemiology and public health unit. There's actually 4 different ways that you can conduct that lab, and kind of like 4 different scenarios, and they love that. So we've got different microbiology students here. We just did an ELISA. So the little blue colored strips that you see down there, that's an ELISA. And then students in our classroom, we don't do the prep. The students do the prep. And so that was just kind of showing that. But yeah, all the labs that Sherry was talking about, Bio-Rad does have really easy, simple follow the directions and very clearly laid out directions. But I also just buy things too from other places and other lab companies and kind of throw together labs. But the students really enjoy the scenarios, that real life application. And when they have buy-in like that, it's really a lot more meaningful for them, and they think on the why a little bit more, and reflecting on that and processing that has really helped, I think, increase their comprehension as well as their retention of what we're actually doing and why we're doing it.
Yeah, very nice. Jamie, any thoughts to add here?
Yeah, the only thing that I would add to this is that for the longest time, my lab reports always involved an extensive conclusion that we break up into three parts: results explained with evidence, and the potential errors and errors, and then practical applications. And I'm always struggling to draw more and more out of the students from that. I took a chance, and with one lab, I went bananas writing a really, really elaborate scenario and dropped them in as field workers where they had to determine the source of an infection for a farmer's soybean field. And the second I did that, and then we had the farm plots, and they were putting this all together, and they were able to use the ELISA to determine where the point of infection was and how bad it's getting based on color changes. Immediately, the amount of information I was getting reflected back to me skyrocketed. So I've been spending a lot of time creating little narratives that go with it that they directly buy into. The narratives that come with the Bio-Rad labs are great, but sometimes it's just a little too remote. And so our school district's kind of surrounded by farmland on one boundary, and there's all kinds of soybean fields out there. They all know about them. But a nasty little disease called sudden death syndrome in soybeans caused by a fungus is just ravaging soybean farmers. And they took that to heart. And so I've been just tweaking labs ever since that. And it's wonderful. They just buy into it immediately.
That's so cool. Yeah, I think it's so important to put these in a context that's relevant to your students. And, that's going to vary from place to place, right? So, yeah, the ag in Ohio, and biomanufacturing in Houston that we've, gone to. We've got biofuels, all kinds of medical, there's all these different scenarios that these skills might apply to. So that's great.
And just one other thing, again, it's make whatever biotechnology that you're doing, make it relevant to your students. Like we've kind of all said, are you more of a medical aspect, and is that what your students are interested in and what serves your community? Are you an agricultural community? Because Jamie has really great resources for like agricultural communities, and that just makes it just such a much richer experience for the students.
Lots more buy-in for sure. Okay, next one. Any memorable projects or activities that your students just killed it on? They just went nuts on.
So I'm not sure if this is still, oh yeah, okay, it looks like it is. So I'm going to highlight something that my students did last fall, and I just mentioned, as did Michelle, the virus tech transmission kit. It was phenomenal. They run a gel, which was fun, and the premise here is that they are given the samples representing 30 patrons in a restaurant plus 2 waiters and a cook. And as Michelle mentioned, there are 4 different scenarios that you could pick. 2 are coronavirus, 2 are noro. I picked the norovirus, and they learn about the chain of infection, which they can tap into lived experience through COVID. They can quickly identify, oh, we could break the chain here if someone wears a mask or if someone isolates, etc. And then they run the gel, they figure out who's positive, and they put that on the restaurant map. And then there are patient history cards where they find out, what did the person eat, did they use the restroom, etc. And then they come up with this, what they think the mode of transmission is. Is it fecal, or is the route to fecal oral, or is it respiratory aerosol? And then the mode, is it direct, indirect, etc. And they just do a phenomenal job. I don't think this is, I had a whole series of pictures that I took from student presentations. I usually don't have students give presentations. This was a case where in their lab groups, I said, I want you to, do this, that, and the other, and show me what you're thinking and why, and then present it to the class. And so it was great because there were moments where other students were like, oh, we didn't think about that. And oh yeah, we got that too, etc. And they had to use evidence. It's a great activity. So cool. I'm going to move forward. since that's not animated anymore. Lee, can you try to move this forward?
I
It doesn't
can. Yep.
seem to.
Here we go. Hello.
Oh, is it?
There. No, it's going. Hold on. There it goes. Okay.
Great.
All right.
So
Sarah?
this is hard for me because I'm trying to single out some things. I have a senior capstone project. So the kids have had the entire junior year to learn these skills, apply it in different areas, and then they come up with a project on their own. I have had everything from 3 students that made a 3D printer that printed a living, a cube of agar with living E. coli in it. I had 3 girls work with an immunologist from Procter and Gamble, and they actually identified an allergen in green coffee that the people hauling the bags of coffee were getting sick, and so sick that they couldn't work. And I had 3 students that transfected yeast with plasmas using nanospheres that they made themselves.
Cool.
You get these kids and they'll want to say, well, can I do this? And I learned very early teaching this that you can't say no, because they will find a way to do it with or without your permission. And when you just start to sit back and say, okay, what do you need? I have that. I can maybe get that. You got to find somebody that's got that to work with. And it has just been mind-boggling, the types of work that these kids have come up with.
Yeah, you definitely have to embrace failure on some of these student-led projects, right? I mean, it's just, and that's real science. So
yeah.
that's, it's a very authentic experience.
Yes.
Okay, Michelle.
Yeah, I agree. And we have our freshman research class that we throw them into when they don't really know a whole lot, but they're trying to figure it out. And then senior year, when they take microbiology, we're also doing a capstone. So the full microbiology curriculum as well as their capstone. And we've actually tried to build some type of like little research project into each one of our courses. But again, they come up with some really crazy things. And most of the time I let them just go with what they want to do. There was a group this past fall where I'm like, what you've just proposed is a senior, like a graduate level thesis project. And had you done your due diligence and research, you would have known that this is not feasible. But that's probably the only time I've put the kibosh on some kids. But just with the labs, I love all the labs. I can't narrow down a favorite. If whatever the lab is of the moment, but the kids in our freshman course, because Jamie was talking about repeat effect pods for conclusions. Well, we do effect also. We've added something, future experiments and questions that they may have.
Okay.
Ooh.
But they love the cheese lab. So we have Ellen Daughtery's book. It's called Biotechnology and the New Millennium. I know Ellen's not actively teaching and has retired from doing professional development, but we still use her tech book and have a lot of her labs based on that. And so we make them incubate the cheese. So they have 15 milliliter conical tubes. And when you're looking in photos, it's like, oh, that must have been the cheese lab, because they have the cheese position. And so they have to, they're like, so some of the older kids call it the armpit cheese.
I mean, armpits make great incubators. You're real close to 37. Yeah.
Pretty darn close. So the freshmen really like that lab a lot, but I love them all. It's fun.
That's so fun. Oh my gosh. Okay. Field trips, and we threw competitions in here too. So any remarkable field trips? I think this is I'm sorry. We're still looking at the cool stuff that your kids have done. So Michelle, take it away.
Yeah, we are really, really, really fortunate to have Mayo Clinic in our backyard. And we have some students of parents who work there. And so for the past 2 years, one of my classes has gotten to go to the sim labs, and they reserve the sim labs for us the entire day. We break our students up into a morning field trip and an afternoon field trip. So it does go till way, way past when school's out. But we were able to go through and do all these rotations. I mean, they're letting my students intubate these $100,000 mannequins and, do chest compressions. We've done ultrasound, aseptic conditions in a clinical setting. This year, they have this new system where you actually got to lay down and every single student got to be hoisted up on the system that they're using now to save their nurses' backs and their health and use those things. So that was really fun. The girl on the left, on the bottom right-hand corner is intubating a patient, and it's amazing the things that they do. Then as the upper right-hand picture, the students who do the research projects, we usually have about anywhere from 35 to 50 that will qualify for the state science fair every year. So those are students from that picture. And then of course, as a HOSA advisor, we are able to take students to competitions. The picture that you see there for HOSA, that's the group of students that we took to Dallas last year for the International HOSA competition. And it makes my palms sweat. And if Amanda, if you're still on here, can agree with me, we're taking 75 to state this year to qualify. So 75 kids in a hotel, 25 hotel rooms for 2 nights. Yeah, I'm not having panic attacks.
and they have to qualify for that. So it's not, just a voluntary thing. So that's amazing. I'll also add, since you're talking about HOSA, that there is a new biotech competition in HOSA that just started last year that Bio-Rad sponsors. So if you do have a biotech program and, you have kids that are interested in doing that, check that out. We can put more information in the chat or talk about it, offline. But, and of course, if you have a HOSA program as well, just know that that's out there and available. It's a nice way to merge those two, health science and biotech into one cool competition. So lots of fun. All right. Next up, we have, what kinds of real world applications? And a lot of you have already talked about some of this to some degree, kind of making it relevant to your students. So any other interesting technologies that you've incorporated or things that are relevant to your students?
I'm going to skip. Yeah, I'm good. Jamie?
What do you think, Jamie?
Just all of them.
All of them. I love it.
Yeah. Where do you start and then when can you stop? There's no way to do that.
I agree. Yeah. You just have to kind of pick and choose what's best for your students and most relevant. We did, this is the 4th year we have a brand new course that we implemented. We only had 5 courses before, but 5 summers ago, I did a research externship for teachers in the summer. And I got to go into an environmental engineering lab that summer. And I was introduced to nanotechnology. So this was Arizona State University, and that's where I did my internship, but it was a collaboration with University of Texas, El Paso and Rice University in Houston. And we got to go to Rice and at the beginning, and actually I didn't know about all this nanotechnology. And because of that, we've put this new innovations course that I wrote into practice. And so we do nanotechnology. I'm doing some biomedical engineering with the students right now. And we do environmental engineering, and then innovations is whatever I want it to be. And then we do CRISPR. So CRISPR is very complex. Bio-Rad has an excellent paper model that we heavily go through first. I do the CRISPR lab with them as sophomores in this class. And then my colleague Amanda does it the next year in genetics with them, but they do the extension where they extract the DNA and then run it out on the gel and say, wow, this construct matches that we really did what we said that we were going to do with the CRISPR lab. So those are innovations that have come up or new things that we've put in place just from our own personal experience and things that we thought that the students should be exposed to.
Very nice. Okay, some of y'all talked on some of this. So what kinds of textbooks or resources or other lab materials that you're using in your class have you found really helpful?
So because of being a CCP program, I have to have multiple textbooks in
Jamie, explain what CCP is.
So
please.
CCP in Ohio is College Credit Plus.
So like CTE in other states, it might be called.
It's just in addition to.
Okay. Is it dual credit?
It's like dual enrollment.
Gotcha.
Things like that. So my kids are in 4 high school credits from me, but they also earn 7 from Sinclair Community College, 7 semester hours.
Nice.
So the number of textbooks that I have in my room is just nuts. But we have the Doherty book. We use that. One of the college classes requires the Seidman textbook, which now is about the size of pharmaceutical desk reference.
Yep.
The Thema textbook is another one that we have to use. But in terms of a really nice, quick book to grab at the bench is really, really cool. It's a little like binder book and it's amazing. Online stuff, we do a lot with New England Biolabs and their web stuff. Bio-Rad, obviously, but one of my favorite resources is grownextgen.org. And if you are at all interested in any ag activities, pound on that website. It is just going to spill labs in your lap.
Nice. Michelle?
So I've listed a couple here. The Ellen Daugherty book that Jamie was referring to is called Biotechnology in the New Millennium. And I love HHMI. That's the Howard Hughes Medical Biointeractives. That's free. And they are really awesome. So if you're looking for just some basic content information on a lot of these things, it's like all biology related. It's like the go-to for AP biology teachers. So I taught AP biology for 5 years as well. And I love using a lot of their resources. Those are just things that I found to be some great resources, but just snagging any professional development that you can go to. And NABT is an organization that I was not associated with before, National Association of Biology Teachers, until I was nominated for an award and won an award. And I'm like, well, I should go and get my award and see what this is all about. Oh my gosh, those are my people. I wish I would have known about this organization at the beginning of my career, and it's amazing. It's extremely collaborative. Love those people and the resources that I've gotten from it and the experiences from it. So kind of like Jamie said too, just reaching out for whatever you can. But you also have to realize you cannot do it all. And we did not build our programs in a year. And you really have to pace yourself and set goals and say, this is how much I can do with my resources and my mental health. And then once I get that established and I feel comfortable, then I can add something new. So don't, just keep in mind, this has been years and collaboration that it's taken to build these programs and courses that we have.
Yeah, we're looking at decades of experience here. So none of these teachers started out knowing all this stuff, right? One thing I will add as a great resource for biotech in particular is Innovate Bio. And someone can put that in the chat, the link, if you don't mind. And they are a consortium of community college, university, and high school biotech teachers specifically. There's a Facebook group or a couple of them. They do webinars and they have curriculum online that you can just download for free. So I'd highly encourage people signing up for that too. Okay, let's see. We're going to go a little bit quickly. So hit the high points here, please, y'all. Any challenges that you faced in your courses and how have you overcome those?
Funding. Funding has been an issue, but it took care of itself after we passed the levy. And enrollment, as other departments introduce other programs, enrollment can be an issue. But because of how my students affect our school report card at the state level, as long as I've got 10, they kind of keep me alive. So, I could go on for days about problems I've had, but in a nutshell, that's it.
All right, Michelle.
Yeah, depending on your funding, like I have here, if you can see the screen, we do have a fee for each one of our courses because it is expensive and you're not, your equipment you have, but your reagents you don't. And those are things that you're going to probably have to purchase, every year, at least some things. And so in the beginning, I found the school district that I was with had an educational foundation and they would accept teacher proposals for money. So I wrote a grant. I know we have two big electrical power kind of water institutions within the state of Arizona, and they offer grants. Sports teams, professional sports teams have grants. Sometimes your educational association, they'll have grants for professional development or for supplies and things like that. So just if you really want this badly enough, you will find a way to make it happen. Just a word of caution is if somebody wants to donate something to your program, I would view it first and make sure that it's something that you can really use.
Yes.
We just had something in our office on Monday and I go, hey, where did this equipment come from? Because it's stuff we would use in our biotech program, but I know it didn't come from me or my colleague. And the computer science teacher said, oh, a parent emailed me and they wanted to drop this stuff off because they thought it'd be really great for us. And I'm like, yeah, this would be really awesome. Do you have the power cords for it?
Oh, no.
There are no power cords.
Yeah,
So
not so useful.
if the parent doesn't come and take it back, it's going in the trash.
Yeah.
Which is really
Oh my gosh.
unfortunate because and make sure like if you've get things from a lab, there are some lab companies that they'll age things out or they replace things or a researcher leaves a lab and they have this equipment and they want to get rid of things. Just make sure it's been properly decommissioned and cleaned before you have that in your lab. So we've gotten some really awesome things, like hundreds and hundreds of dollars worth of things before donated from those situations. But just check it out and be cautious before you accept those freebies because they're not always free.
Yeah, no joke. Some other things that I've gotten ideas from other teachers who've been in this, those small local grants like Michelle was talking about, sometimes even like your grocery store, PTO, there's all kinds of community organizations, Rotary, things like that you can check into. And even if they don't necessarily have a scholarship, sometimes you can still approach them and ask. So we've had teachers that have had good luck doing that kind of thing. And they're the only ones asking. So they get the grant, or they get the money. And it also helps sometimes to have a particular target. Can you find gloves for my students this year, for example, or something like that? So those kinds of tricks can help you get those little grants. And then, once you get little grants, it's easier to get bigger grants. It kind of builds and builds and builds. And we have a whole grant writing webinar. I'll have to find that, and we might want to redo that now that I'm thinking about it. But we can address some of those tips and tricks in a future webinar for sure. Okay, next one. Let's see, what have we got?
And
next?
just you can quote me on my little quote that I have down there. I always tell my students this, and you need to take this
So
to heart
true.
too. And I do too. Sometimes I'm like, oh gosh, I need to preach to myself. The answer is always no if you don't ask. And it might
That's
be no,
golden.
but
Yeah,
it's
that's
always
true.
no if you don't ask.
That's exactly right.
So this is a
Okay.
quick slide. Is there anything, Jamie, you want to add? Oh, never mind. Okay.
Oh, sorry. Did I skip one ahead?
It's okay. Go ahead.
Yep. Any success stories from your students?
So
Highlight.
this one is just one of many. When we were writing, we had a gentleman that was in charge of our consortium, in charge of making the relationship stick. He is as old as dirt and he is in failing health, and he had a complication due to a new medication and ended up in the emergency room. And as this doctor's working on him, he is talking to this doctor. We're like, okay, so how'd you get your start? Because he's always trying to figure out ways to make everything better. Turns out the doctor that worked on him and ended up saving his life graduated from my program.
That's amazing.
And so that one's really special to me.
That's pretty cool. Very nice story. Okay. How do you stay updated on advancements in biotech? PD, what other stuff do y'all use?
Okay, this is me. I'm just gonna we have 2 minutes, so I'm quickly just say 2004. I was able to go to the Biotech International Conference. It was in San Francisco. What was interesting is people were protesting GMOs. I could tell you a whole thing about that, but that becomes interesting now, even in my class, to go back to that. And then I won't even talk about this, other than you see the windows there, the 4 colors of DNA. That's the Institute of Genomic Research,
Cool.
Tiger Building number 5. Okay, so that comes up in class. So I give students I call it kind of my nerdy presentation of things I've done that lend itself to to the class. So this is Eccles Institute, University of Utah. DNA staircase. I thought was my favorite staircase in the world. And I met Dr. Frank Kopecki, knockout mouse, Nobel Prize. And then here I am. I wrote a grant to go over to England. I saw Darwin's house. This is outside of Watson Crick's lab. There's his model. And then I met James Watson a few years later. And so then here I'm doing my jazz hands, as I tell them, at Yellowstone. Why? Well, and that's my very first selfie that I couldn't resist. True, I was late to the selfie game. That was a brand new sequencer, but jazz hands because of PCR and Taq polymerase. I was gold medalist in micropipette Olympics, so I showed them this sort of thing. Call this the holy grail of selfies. It's Paul Anderson, Sean Carroll, and Neil Shubin, and it's at the Rock and Roll Hall of Fame. Rock and Roll Hall of Fame, and that was an NABT. Okay, so all right, so I won't go on, but I actually want to say this. Michelle brought up HHMI, and I want everyone to know that if you don't, one, they started an online community last fall that you can join in a lot of different groups like biology teaching, et cetera, science and equity. But they're going to accept applications. I'll go back one. Accept applications for their brand new cohort, Ambassador Academies. The applications opened in March. It's a 3-year commitment, but they pay for everything. It's amazing professional development. In the past, you were able to go to Mecca Nerd Camp at HHMI. They pay, again, phenomenal. So I'm putting that out there.
That's awesome.
And then the next one I wanted to tell you about 3D molecular designs. I have a mini tuber here, which is awesome to use. But here I am at a work session, but they are going to have now you do have to pay for these, but Modeling the Molecular World, it's in Milwaukee, Wisconsin. And that's an incredible workshop. And then San Diego Zoo you can do this for free. You have to apply with a partner, someone else in your building. Erin, that's the person standing with me. This was 2018. She's AP environmental science teacher. And we realized 50% of her kids take biotech as seniors. And so what's the overlap? What can we do? She's obviously teaching conservation. What can I do, et cetera? And you can, if you want, you can like pay to reserve like $2,000. Otherwise, if you're selected, they'll pay for everything. So I encourage you to do this. I learned a lot about conservation there. I'm holding an elephant tusk. And they told us that the bullets were shot from above. So this was seized at the border. So they were shot from helicopters. Anyway, that application is open, I think, until April 7th. So I highly encourage that. And then webinars. Webinars are free. And, now since COVID, these are becoming better and better. So highly recommend that. Okay, go ahead, Michelle.
And just since there's so many awesome resources in here, we're definitely sending this out. So don't worry about trying to write all that down. You'll have the PowerPoint you can refer back.
Well, Sherry, I want to get on your professional development team and just tag me along and bring it some space.
For real.
Those are awesome. But I got to meet one of my heroes. He saved me teaching AP biology. Paul Anderson was at our state science conference this year. And so that was just amazing. But these pictures are in here, just professional development, as much professional development and as much free professional development as you can find is really, really awesome. Find your people. Find people to mentor you, find people to coach you, find people. I'm the kind of person, I need the little kick in the hiney to get myself going because I'm intimidated. I have imposter syndrome like my students and never feel like, oh, I can't do that. I'm not good enough for that. I don't know how to do that. And I need those people in my life that push me to do things. And then I just did with our state science people, I picked this book up this summer. I found it in a pile of things. I finally had time to go through it. I'm like, oh, this looks like a good book. And I started reading it. And then I found out there was a book study happening on it. And so I joined one module of the book study on ambitious science teaching. And it's been really great. And meeting more people throughout the state and just making those connections. It's really important.
Very nice.
So
go ahead, Jamie.
Everybody's talked about PD, so I'm going to leave that alone, but put an exclamation point after it. It's really, really important, and there's so many options out there. My daily routine starts with about an hour of reading a morning, and I jump on various websites, and I just start to read, and I'm just trying to stay abreast of what's happening. We're in the midst of the one science that is changing faster than any science in human history. And so by reading, I just feel like I've got a half a chance to stand in front of the kids.
Yeah, really important. That's great. Let's see. Any thoughts here about how are you preparing your students for, what's next in biotech?
Same thing, read and
Yeah.
professional developments. As long as you know what's coming and what's happening, the techniques will transfer in. You just got to know what's there.
Yeah. Let's see. Okay. This is a great one. What advice would you give to new biotech teachers? So if they're just starting or considering, sorry, go ahead.
Michelle mentioned this. So Eli Lilly is in Indianapolis. They do tremendous work with the community, but they have teacher giveaways of equipment and so on. So if you have a university or so on, but I totally agree with Michelle. Make sure you know what you're getting and don't like someone offered me a DNA sequencer. I said no. And like, well, why would you say no? Because I am not even going to begin to pay to maintain that, et cetera. So some things I just don't think are worth it. Invite your administrator in. This is my principal. Now you can tell he's just learning, not the greatest technique, and the girl is kind of like Mr. Van Lambrook. But invite in your administrators. Have your students teach them how to micropipette. Stay organized. So this is a Bio-Rad box, and you can see, like, I'm not relabeling wash buffer every single year. I save those. The same thing. I've got an antibody detection. I got all those tubes in a bag. My antigen detection. So do not relabel every single year. Some things you have to, like some things you have to worry about enzyme and so on, but other things you don't. Here, my lab is organized. So what you're looking at is when you walk in my door. I've got a class supply area, so hot pink rack. You take anything off the hot pink rack, it always comes back. No one else in the class has that hot pink rack, only mine. It's labeled, it comes back, and then I can refill it for my next class. And then you see where it's like PCR tubes. So I empower my students to know where, and I suspect Jamie and Michelle do the same thing, where things are located so they know what to get at certain times. Okay, stay organized, stay organized, stay organized. So here is a side of a gel chamber. That's group 1. You can see that's my staining dish. If I do staining the gels and I'm not using UView, I've got staining dishes. I've used those for years. Then the destaining dish, and I have 5 sections. So, I label them so I know it's group 1 in whatever class period. And then have fun. So, you want to do Charlie's Angels with me with micropipetters and it's Blues Week. Sure, I'll stand and pose with you. It's Senior Week and you want a picture at our lab. Yeah. You're at prom and you want to do PCR and send it to me. I call that a win.
That's awesome.
Then, yeah, I love this picture. They didn't know I was taking it. This was several years ago. But what you're trying to do, right? And I know all of us as science teachers cultivate community labs, right? Working in lab group. You see the boy on the left is holding the other boy's arm.
Yeah.
He's trying to load a vertical gel, right? And the other one is totally, they didn't know I was taking the picture. I love that. Again, here that I call this micropipette art. Maybe the last day of the school year, maybe going to winter break, you've got a little time. The wax paper, different colors, and they use micropipetters of different volumes, and they can, make different things. She put, I love biotech. Here, I say vertical gels. I started doing this in 2014. I tell my students that your greatest memories are going to be with your lab partners. And so I say, I take a picture like in April. I said, pose any way you want. And then I surprise them on the last day with the picture. And then a vertical gel, I save those and then I wash them out and then put magnets on the back and then they put on their dorm fridge. And so I've gotten a lot of kids.
I love that. That's so cool.
I would just say, just do it. You don't have to know everything. Honestly, don't tell anybody else, but there's some pieces of equipment, like the more techie things that you have to program. I don't know how to do all of that, but I have the kids do that. And like one year when something happened to one of our really expensive pieces of equipment, I go, here's the tech support number with Bio-Rad, get on the phone with them and figure it out. And they did. And now he's like at a professional research lab. So I'm going to credit that for, helping him on his research journey. But you don't have to know how everything works. You can figure, and that was a big thing for me, as getting over having to know everything and thinking I had to be all and do all, and just being willing to fail and to just go in there and go, yep, that's science. Let's figure it out together and give up some of that control. That was really freeing for my career to be able and to model that for the students too. Yeah. That things don't always go as planned, but let's figure it out. Let's find a way to make it work. And then the other thing, oh shoot, it came and it went. So just do it, try new things and find those mentors. And that's, I think, really, really important.
Mentors are really a big deal.
Oh,
Yeah.
here's what I was going to say. I got this from the person who came before me. Have elected positions in your classroom. We have
Ah,
resource associate, and it's a big deal. Like they have to run, they have to give a speech, they vote on each other and who's going to do this. So our classroom resource assistants, they keep track of the absent people and they put the names on the papers and they email them what we did and they help them when they come back. They check in with them. We have a safety officer who gets the SDS together, and we have a folder in our drive that the safety officer is in charge of putting all the SDS into advance of the lab. So the students have to have that safety in their things. They make sure that everybody's wearing the goggles and the closed-toed shoes and their hair's up, and they run the goggles cabinet at the end of the class. And then we have media techs. Media techs are the best. They have
I
to
need
have
one of those.
they have to be free either before school or after school, so they can't have a late block or a zero block. And they come in before the lab. You don't label all the tubes and make up the reagents. They make sure that the lab soap is always available and the Lysol and the 10% bleach. And they have to keep all those things supplied and do the labeling and the pipetting. And you just kind of watch over and check their lab math.
That's
Thank me later.
amazing. That's amazing. I love it.
So
Jamie, anything to add here?
yeah, I've got three things. These tend to be a little bit more philosophical. First and foremost, and I'm just going to skip to the last line of this, find a hobby that will allow you to divorce yourself from biotech on weekends and for some time in the summer.
Yep.
This science will eat you up, and you've got to be able to push back from the table. The second thing is that you've got to go into this knowing the windmill factor. There are things you cannot control, and they will go wrong, and that's fine. Deal with it, but you're not going to have time to lance at windmills. Last but not least, this too shall pass. If things are going well, this too shall pass. And if they're going bad, this too shall pass.
Great advice. Okay. Thank you guys so much. Now I'm going to just let y'all unmute, or if there's questions in the chat that came up, but were there any burning questions that we want to cover in the Q&A?
There were a couple, so some of them were addressed, but I don't know if there are anything else we want to say about, let's see, there was a fun question about, have you had any incidents or injuries? If so, what was the worst of the worst?
That's a good one.
And then Heidi was also asking about ways to integrate biotech into a microbiology curriculum, and just more ideas also about professional development. And now we have, so we've got a range, and two others that have just popped in about cloning and sequencing and grading. So does anybody want to pick one of those four?
So the other one was, has anyone used cloning and sequencing, and how do you deal with grading in your classes? That's a really great one. Do any of y'all want to address any of those?
I do some, the cloning work essentially is the Piglo lab. And so most of us do that kind of cloning. As far as like plant cloning and plant cell culturing or any cell culturing, I tried that. All I did was grow copious amounts of mold. So we've walked away from that, and I spin things differently. As far as sequencing, I don't have a sequencer, but we do 1 or 2 sequencing labs a year, and we send stuff off to be sequenced, talk about the process and the results, and do a lot of blasting.
We also do regarding cloning and sequencing. I'll put a link in the chat. We just recently had a webinar. Lee, do you think putting a link to that webinar would be useful?
Yeah, we have the cloning and sequencing. We have a whole cloning and sequencing series, and so that there was a webinar on that just a couple of months ago, and so Yolanda can do that. So that's a great way to find out more about that one. Any thoughts on, let's see, grading was another great question. How do you handle grading lab reports, or do you do lab reports or lab practicals?
I think Michelle and I probably are I think we all do lab reports of some sort, and it's grading a lab report. I'm not exactly sure how else to look at it. I kind of pace mine out though. I focus a lot on the structure of the lab report the first quarter of junior year. Then second quarter, once we've got the structure down, then we focus on the results section of the conclusion. Then we bump it up into the errors third quarter, so that everything just kind of maxes out by the time fourth quarter rolls around, and they're really focusing on everything, including potential applications for the technology.
Awesome. We're almost at time here, so I want to be respectful of that. We'll probably stick around a couple more minutes, but I did want to, if you don't mind filling out our survey, this will allow us to contact you if you have any questions. We'll send you the PowerPoint and the recording of this as well. So if you can just hit that with your phone, and I'll also put a link in the chat as well. So give me just 1 second for that. So here's the link for the survey if it's easier to do on your laptop. The other thing that I wanted to mention was that we do have a Bio-Rad textbook. And so this is, if you'd like to request, if you're thinking of starting a biotech program, and you'd like to take a look at the textbook, just let us know. You can put that also in that survey link. There's a couple of fields where you can fill out, other or whatever comments you'd like. Or you can just send me an email. You all have my email now, and we can keep you up to date. And lastly, the other thing I wanted to point out is we have a whole list of classroom resources. I don't know if that's going to work. Yolanda, do you mind reformatting that for me? The Zoom formatting doesn't let you you have to have the whole HTTP thing on there. Classroom resources that are available, and those are a good start. And I think that's it. I'm going to thank you, panelists, Sherry and Michelle and Jamie, so much for your time. And like I said, I'll be around here. I'm just going to stop the recording now, but I'll be around for a little bit to answer more questions. And other than that, thank you so much for your time. Appreciate that.
Thank you for being here.
Absolutely. Okay. Stop.